the stem cell derived spermatogenesis for management of male infertility, which is one of our recent works. All right, so I'll start by introducing Professor Islam Al-Dababsi, on whose behalf I'm presenting this topic today. So he's the director and the founder of ID Stem Cell and Genome Institute, as well as the founder of the ID Medical Group with ID Clinic, ID Hospital, and ID Kids, which is a center for kids with special needs. To begin with, he's a professor of genetics and stem cell researcher for the past since 1999, actually. He has a PhD in cardiovascular surgery. He's a member of ISSCR and associate of molecular and cellular therapeutics, and has several publications. This is our team in ID Stem Cell and Genome Institute. We have our own GMP laboratory with the hospital and an IVF center. We focus on fertility treatment and several trials for management of different diagnostics and different diseases that can be treated with stem cells. So the objectives of today's presentation, we will discuss the impact of male infertility on the population. What are the available treatment modalities for male infertility, as well as stem cell therapy as a potential solution for male infertility. And then we'll discuss the key findings from the recent researches and the key findings from our research, and discuss what is the process in details. So what is male infertility and what is its impact on the population? So as defined by the World Health Organization, as male infertility is the inability of a male to make a fertile female pregnant for a minimum of at least 1 year of regular unprotected intercourse. So the male is solely responsible for about 20% of cases and is contributing a contributing factor in another 30%, 30 to 40% of all infertility cases all around the world. Specifically in the MENA region, where we focus our clinical trials on patients from MENA region, infertility rates in the MENA region are well above Central Europe, Western Europe, and Southeast Asia. So in the MENA region, there is 800 infertile men per every 100,000, which is quite high statistics. So out of the 3 countries that presented with an increase in the trend of male infertility, 2 are from the MENA region, Turkey and Morocco. That is why it is very important to focus on other modalities of male infertility management other than the ones that are already present. So to know how to treat male infertility, we need to know what are the causes. So what can lead to male infertility? First, it can be congenital. So a congenital urogenital abnormality. So the patient will be born with no ability to produce sperm. Another causes can be endocrinological, such as well as congenital syndromes and acquired syndromes. Obviously, environmental toxins, genetic causes such as Klinefelter syndrome, Sertoli cell-only syndrome. Some idiopathic causes are there as well, which we can find the reason for. Immunological causes, hypophysitis and hemosiderosis, hemochromatosis, obviously malignancies, post-treatment, post-psychotropics, post-exposure to chemotherapy or radiotherapy, as well as some reasons of sexual dysfunction, such as premature ejaculation, anejaculation, and infrequent sexual intercourse. And frequent urogenital tract infections can be one of the other causes as well, causing permanent damage to the male anatomy. So what are the available treatment options right now for male infertility? Obviously, hormonal therapy. So replacement of the hormones that help the male body produce sperm. So hormonal treatments can sometimes stimulate sperm production, especially in cases related to low testosterone levels, but hormonal therapy does not work for everyone, obviously, particularly men with non-obstructive azoospermia or azoospermia in general, caused by genetic factors, any congenital conditions, any post-chemotherapy or post-drug exposure. Another available treatment option is surgical sperm retrieval. So surgical techniques like testicular sperm extraction is used in azoospermic men to retrieve sperm directly from the testes. It is considered successful in some cases, but it is still invasive and expensive and offers no guarantee of finding viable sperm. So in patients with low testosterone and low quality of sperm, even a surgical approach will not guarantee fertility. Another techniques are assisted reproductive technologies. So IRTs, like in vitro fertilization or intracytoplasmic sperm injection, is the most common treatment for male infertility. However, they're expensive, invasive, and the success rates are very variable. So additional to that, IRT does not resolve the underlying cause of infertility. It only helps facilitate conception. But if the person is azoospermic, he remains azoospermic even if he did do an ART. And obviously, we have an option of having donor sperm, but this is where an ethical issue arises. And not everyone, not every family would be okay with having donor sperm and conceive through a donor sperm. In the end, there is an ethical issue and there is a psychological impact that might affect the family status and quality of life, especially in the MENA region. Many cultures here and many people and families will find it unacceptable, actually. So what are stem cell therapies and why we consider them a potential solution for total infertility in males and females, but today we're focusing on only males. So stem cell therapy is becoming very, very popular lately, as we can see by these multiple conferences regarding stem cell approaches. So it is a groundbreaking approach to addressing male infertility. Specifically, MSCs, which are mesenchymal stem cells, they have drawn attention for their regenerative abilities and the potential to restore sperm production. On the other hand, we have iPSCs, which are induced pluripotent stem cells. They offer also a promising treatment option because they can be reprogrammed and develop into any type of cells. And in our case, if we want to treat male infertility, they can be reprogrammed into sperm, right? Or a sperm precursor cell. So let's review some types of stem cells. So what are mesenchymal stem cells? MSCs are adult stem cells found in various tissues, such as the bone marrow, the adipose tissue, and umbilical cord blood, as well as the placenta as well. These cells can differentiate into several types of cells, including bone, cartilage, and muscle cell. However, the process is timely and takes a lot of effort and knowledge and specific techniques of cultivation. The other type that I wanted to speak about today are induced pluripotent stem cells. So iPSCs are lab-generated cells. So they are created by reprogramming an adult cell. So a skin cell, a blood cell, an adipose cell, same as an MSC, they're being reprogrammed back into an embryonic-like pluripotent state. And after that, putting them in different types of mediums will allow them to differentiate into any type of cell, including sperm cells. So in the treatment of male infertility, iPSCs offer a promising approach, especially for cases where patients cannot produce sperm naturally due to genetic or developmental issues. So by differentiating the induced pluripotent stem cells into sperm precursor cells, researchers aim to enable the production of functional sperm that could be used for assisted reproductive techniques. However, there is not many studies yet on induced pluripotent stem cells developing into a sperm study. Actually, there is none yet that have had a successful fertilization of an embryo. So let's dive into the basics of human spermatogenesis so that we understand the background of our study. So spermatogenesis is a tightly regulated process by which the male germline produces mature spermatozoa from undifferentiated stem cells. This was very important for our study, and this part of anatomy and physiology is very, very important to understand the pathophysiology of each disorder for people with infertility, especially males with infertility. And then later this anatomy and physiology will help you understand why did we choose the approach that we did and how did we come to this idea. So the total duration of spermatogenesis is approximately 64 to 74 days, and it is divided into three main phases: the spermatocytogenesis, which is the mitosis, the meiosis, and spermiogenesis, which is the final differentiation of the cell. So the first phase, spermatocytogenesis, is primarily a mitotic phase that begins with SSCs, which are spermatogonial stem cells. They're located in the basement membrane of the seminiferous tubules, and this process lasts approximately 16 days. So the mitotic phase is 16 days. These stem cells undergo asymmetrical division to both self-renew and produce differentiating type A spermatogonia and type B spermatogonia later. So after the successive mitotic division into type A spermatogonia, they divide and rise into type B spermatogonia, which are committed to entering the differentiation pathway. So type B spermatogonia then differentiate into primary spermatocytes, which are the first cells to enter the meiotic phase. Not all of the spermatogonial stem cells during the spermatocytogenesis will develop into type A, and then from type A into type B. So if in the beginning we have around 1 million stem cells, by the end we will have only around 600 to 700 primary spermatocytes. So I wanted to pay attention to the molecular markers that will help identify the progression through these phases. So in each slide, I will be explaining what are the main markers that will help you identify whether this cell has been progressing through the mitosis to meiosis. So specifically for the mitotic phase, we have such a key molecular marker that is expressed. It's the KIT, c-KIT. That is a receptor tyrosine kinase that indicates commitment to the spermatogenic lineage. So when you have the cell expressing KIT, you know that it is differentiating into a primary spermatocyte eventually. And then another marker is the STRA8, stimulated by retinoic acid 8. It is another critical marker activated by the retinoic acid that initiates entry into the pre-meiotic DNA replication and is considered a hallmark of differentiation from the mitotic to the meiotic phase. So first, we need to identify whether those cells are growing in the right direction by expressing KIT, and then we check whether they are progressing well with the STRA8. So following the mitotic expansion, primary spermatocytes that are formed by the end of mitotic phase will now enter the meiotic phase, which will last approximately 24 days. So the meiosis is divided into primary and secondary meiosis. So primary meiosis, in the primary meiosis, primary spermatocytes undergo recombination and homologous chromosome segregation, resulting in secondary spermatocytes, which are haploid in chromosome number but still contain duplicated chromatids. So these secondary spermatocytes rapidly proceed through meiosis 2, which will produce round spermatids, each containing a haploid set of chromosomes. So what I wanted to pay attention to in this slide is that in meiosis, we will have a haploid set of chromosomes, which is very important to notice to avoid any chromosomal abnormalities further within the sperm. So one of the hallmarks and one of the key markers here is the gamma H2AX, which is a phosphorylated histone variant. It signals sites of double-stranded, essential for homologous recombination, double-stranded DNA. So in addition to that, we have DMC1 and RAD51, which are recombinase enzymes involved in the repair of these breaks and the alignment of homologous chromosomes. So in case the gamma H2AX has detected any breaks in the double-stranded DNA, DMC1 and RAD51 will be there to fix it and confirm it and fix it, and then further make the cell, help the cell progress further through the meiotic phase. So these markers are often used in both research and diagnostic settings to verify that the progression through meiosis is normal. The third phase, the last phase, the spermatogenesis, is a post-meiotic transformation process that lasts also around 24 days. So during this phase, the round spermatid that's formed by the end of second phase of meiosis, they undergo morphological and structural changes to become fully differentiated, elongated spermatozoa. So during this phase, there are several key markers and several key processes that need to take place. So first, we have the acrosome, which is a Golgi-derived vesicle. It shows the elongation of the nucleus with tight chromatin condensation. It is a nucleus with tight chromatin condensation, and it helps the development of the flagellum and shedding of excess cytoplasm as residual bodies. So the chromatin remodeling is a central event in spermatogenesis, driven by the sequential replacement of histones with transprotein 1 and 2, as well as protamines 1 and 2. So this allows for further packaging of the DNA, essential for the protection and delivery of the paternal genome. So let's summarize this slide, and in more simple words, we need, for the sperm to be fully functional, we need two parts, acrosome and the flagellum. The acrosome will contain the tight chromatin, and the flagellum is there for the cytoplasm and the residual bodies. And the key proteins here are the transproteins, transaminase proteins, and protamines. So all of this helps normal DNA to form within the sperm, and the defected DNA to form within the sperm, for it to avoid any genetic abnormalities further. So the acrosome is critical for oocyte penetration. As we know, it expresses the protein acrosine and SP10, which are also hallmark markers for normal development of the sperm. Another cytoskeletal and structural protein like tectin and ODF2, outer dense fiber protein, they're key for proper flagellar assembly and motility. So all four of those markers need to be tested in order to know whether the sperm is fully functional and of good quality or not. So now we have passed the three phases of development of the sperm, spermatocyte, but where they are morphologically complete, but still for the next 10 and 12 days, they will develop progressive motility. For this, we have markers such as CRISP1, cysteine-rich secretory protein, which plays a role in zona pellucida binding, and P34H, which is involved in sperm egg recognition. So this is just an additional markers that will help you identify whether those will be able to fertilize a female egg. Okay, so what is our innovative approach on the management of male infertility after we've discussed the normal physiology? So this is technique mainly sponsored by the amazing brain of our professor, Dr. Islam Dababsekh at ID Stem Cell and Genome Institute. So this information is obviously patented. So I would like, you can remember it, but please no pictures, and do not share it with online and publicly. So what is the protocol of our MSC injection? So first, when we get a patient and we confirm that he has non-obstructive azoospermia, he have tried to conceive, and there is a confirmed diagnosis of male infertility, first what we do is consent, obviously, all ethical issues resolved, and we give him a GCSF factor administered into his blood to stimulate the bone marrow, to mobilize MSCs that are in his bone marrow to go into the peripheral blood. After that, we get 300 mLs of blood from the patient, filter it to isolate MSCs, and keep it in approximately 2,500 international units of heparin per 100 ml added for anticoagulation. So the isolated MSCs, we then further mark with CD105, CD73, and 90, which are the standard markers for MSC identification. And then under microscope, we examine them to confirm that approximately at least 95 to 98% are viably in polymorphic nucleus containing cells of various stages. So in the end, the cells were concentrated into 2 milliliters portions by centrifugation. So by this time, every portion, every vial has around 95 to 98% viable polymorphic MSCs. After that, patients undergo IV anti-aging program with the same MSCs and some intratesticular administration of the MSCs as well. So what is the protocol of sperm cultivation? How do we grow sperm out of his own blood cells? So this research begins with the administration of granulocyte colony stimulating factor, like I've mentioned before. After that, the blood is withdrawn. We mark it with CD105, CD73, CD90 surface markers. Additionally, it is then stained with CD133, which is a marker commonly associated with an embryonic origin, and pluripotency, which aids in identifying stem cells with higher regenerative potential. So why do we need this? Is because CD133 identifies whether the cell has the potential to be pluripotent, has the potential to grow into anything, so that we do not need to reprogram it into being an induced pluripotent cell. It is already pluripotent by its own nature. So after this analysis, approximately 20% of the CD105 positive cells also expressed CD133, which is quite low of a number, but then after cultivation, you still get the same result. So this is our MSC pool. This is a picture of the MSC pool identified with the CD105 and 73 and 90. All right, so what is step 2? Step 2, further, the isolated MSCs are also tested for OCT4. Why OCT4? Because it is a marker of pluripotency. It is necessary for maintaining stem cells in an undifferentiated state. We want them to remain in an undifferentiated state to then be able to manipulate their growth into whatever cell we want. So the same technique that is used with induced pluripotent stem cells, but without reprogramming and damaging the cell, we get the cell by itself from the blood. So 75% of the CD105 and 133 positive cells also were positive for OCT4, highlighting their potential to differentiate into gametes, particularly spermatocytes. So this property indicates a possibility of those cells to be germ cell inside the human body, which is very controversial, have been very controversial, because the books state that there is no germ cells inside the human body, an adult human body. It is a hallmark of an embryonic cell. However, several markers can identify that those cells do exist in very less quantity, but they do exist. So step 3, we cultivate the spermatocytes. How do we do that? So further, the isolated CD105, 133, and OCT4 positive cells, they are cultured in a specially designed environment that mimics the embryonic germ cell conditions to promote their differentiation into spermatocytes. I will not be mentioning what it is exactly, because it is a patented medium made by the ID Stem Cell and Genome team. The culture process spans several days. So we try to mimic the same spermatogenesis technique that is there, that happens naturally in the body of a fertile male. So approximately by day 13, we noticed that those cells started expressing dazzle. Dazzle is a marker essential for the initiation of meiosis, indicating the transition of the cells from diploid to haploid states. As I said, in the normal spermatogenesis, having a haploid state of chromosomes is very essential for the further development of the sperm. So this is obviously a key milestone in the development of sperm in vitro. So this is our MSC up there, the 2 pictures of MSC pool staining with OCT4, and below those that stained with dazzle on day 13. After that, on day 15, the majority of the cells that were already positive with dazzle, they also started expressing DMC1, which is, as I mentioned before, it's a marker for homologous recombination during meiosis. So the presence of DMC1 obviously confirms that the cells have successfully undergone meiosis. It's done. Both 2 phases of meiosis are done, and now we have a primary spermatocyte. So this resembles and suggests that the cells originally from the patient's bone marrow have the potential to develop into functional gametes, which is also supported by the theory that both the reproductive and the hematogenous systems originate from the mesoderm. So because they originate from the mesoderm, they can be developed into the same kind of cells that they develop in vitro. They can be developed into the same kind of cells that they develop in the human body naturally during the embryo stages. After that, we have on day 32, we have acrosine, which is a protease enzyme that is crucial for spermatogenesis. And those cells were stained with acrosine to identify the formation of the acrosome, which is a dense chromatin granule. So on day 32 to 45, the acrosine granules were only becoming more concentrated, and they would drive from the center of the cell into one pole. So by day 45, we have already an acrosome within the cell, and now they start developing the flagellum and display some motility by day 45, which only resembles the same technique and physiology of spermatogenesis in the normal human body. So now we have a fully developed gamete with an acrosome and a flagellum, and we extracted them from the cell culture, and they exhibited distinct heads, acrosomal bodies, and tails, marking their readiness for fertilization. So the normal spermatogenesis, as I mentioned before, takes around 64 to 75 days. But here, since they have already developed motility on day 45, that means that they already can cause fertilization if they're introduced to an egg, because all the essential structural bodies within the sperm are already fully developed, and they're even motile. So these pictures show MSCs marked with the MC1, and this is the acrosine granules shattered all across the cell, and then some of them, they are concentrated by the pole, if you can notice it. And some of them, they're already concentrated, and some of them, they're still shattered all around the cell surface. Okay, so after that, we've attempted an IVF by a sperm that was fully made in vitro. So the mature gametes then used for IVF procedures with donor oocytes. Obviously, consent was obtained from all parties. During the acrosome reaction, acrosine facilitates the penetration of the oocyte zona pellucida, which resembles the normal physiology of the body, leading to successful fertilization. Initially, the thick membrane surrounding the spermatocyte prolonged the fertilization process, but after further cultivation, the membrane thins, significantly speeding up the fertilization. So the more you leave it in the culture, you let it cultivate, the thinner the membrane becomes, and the more easy it becomes for the acrosine to shatter and fertilize. So after that, we left some of the cells until day 75 to undergo full spermatogenesis, just like in the normal human body. But what we notice is from day 45 till 75, which is approximately a month, 30 days, they would only develop more motility and grow their tails, the flagellums. They would only grow that, which actually we noticed that it made them a little more fragile if we wanted to extract them from the medium later for fertilization, which is now controversial whether it is needed to maintain the 75-day spermatogenesis, or is 45 to 55 days enough for it to be fully functional motile sperm. So these are the mature gametes inside the MSCs. And there is a video. I'm not sure if I will be able to play it. Yeah. All right. So the video just showed the same picture that is here, and an egg, a female egg, and how the sperm from one of our cultures moved towards the egg, and actually was able to break the membrane and fertilize it. So obviously, to maintain the ethics and of all of our consent and promises to our patients, the fertilized embryos were dissolved before reaching the morula stage to not break any rules. So here, let's identify what are our future research and clinical applications and what is the further approach that we need to maintain. So obviously, we need more clinical trials for assessing the efficacy and safety of such approach of treatment of male infertility. And we need different protocols for different cases of male infertility because there is no just one specific type of patients that can be treated by the same protocol. We're all focused right now on regenerative medicine and on precision medicine, which identifies the individuality of each person and the requirement to treat them with a specific approach and a specific technique tailored to their own body. So I wanted to extend a special thank you to our partners all around the world, all of our sponsors, all of the amazing, amazing professors that help us with our ideas and develop our research. That's the list of our partners. And thank you as well for listening.
Thank you so much, Islam, for the wonderful presentation. And we are now open for the questionnaires. Anybody have any questions? They can just raise their hand and we'll bring them up. Any questions? Okay. In the meanwhile, we'll be launching the poll. Participants are advised to participate in that poll, please.